Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@eapen.info. My guest today is Professor Tudor Oprea, who is Professor of Pharmaceutical Sciences and Chemistry and Chemical Biology and Division of Translational Informatics at the University of New Mexico. His current research is in the Department of Validated Artificial Intelligence Models for Target Selection and Drug Discovery by combining numerical and text-mined information, model human via knowledge graphs. Welcome, Tudor.
Thank you, Jill, for giving me this opportunity to introduce people to the exciting work that we've been doing.
Great. Yeah, thanks for doing this. I want to start with one of your recent papers entitled Exploring the Dark Genome, Implications for Precision Medicine, in which you say the increase in the number of both patients and healthcare practitioners who grew up using the internet and computers, so-called digital natives, is likely to impact the practice of precision medicine and requires novel platforms for data integration and mining, as well as contextualized information retrieval. Now, we have seen interest in the use of artificial intelligence in healthcare increase over the last decade. And increasingly, there are applications that work on EMR data and other types of data, both structured and unstructured, in the healthcare provision side. On the pharmaceutical side, I don't know, maybe you can tell me, my experience has been that they're a little behind in the application of these technologies. And so you want to talk a bit about the paper and what you're doing in this area?
Absolutely, Jill. Thank you for mentioning parts of my work. So essentially, I start with the observation that digital natives, which I typically define people who were born during the age of internet, and some of them, of course, end up being healthcare practitioners. The other ones end up as patients, or at some point, all of us will end up as patients. And so what's happening is that there is a lot of information that's pretty much assaulting us across multiple channels, not just television, at least if you're in the United States and you watch TV, almost every 5 to 10 minutes there is a commercial that discusses some novel drug and it's how it would possibly help you if you suffer from a so-and-so disease. So that's one channel of communication. Of course, everybody goes to social media, Facebook, LinkedIn, other platforms, and Reddit. And pretty much we have a lot of information that's coming our way. So what I try to address in this paper is if we were to try to develop tools that target the digital natives, how would we try to make those tools look like? And of course, I'm in particular interested in ways to provide information that is distilled primarily addressing the biomedical scientist, but also the clinician and hopefully the patient as well. And in doing so, we have developed this platform, which is available at the nih.gov website. It's called Pharos, and that's spelled with a PH, so it's pharos.nih.gov. And that platform is a data aggregator that includes information about proteins, diseases, and ligands. And by ligands, I mean chemicals that also includes drugs. So what we discuss in this paper is if I were to find a way to quantify data availability and try to take information across all these channels that provide information related to proteins, I would probably discover that there are some proteins that are really well studied, but there are also some proteins that are understudied. And we have proposed a knowledge-based classification system for proteins. We divide them in 4 categories. And the take-home message of this is that a small fraction of the human proteome, about 600 proteins out of 20,000, are what we would consider drug targets. In other words, we know with reasonable degree of certainty that's how drugs work. Also, at the same time, about 1/3 of the human proteome, about 6,500 such proteins are what we call T-dark. And those are proteins that are understudied. So in this paper that we are discussing, I'm trying to look at the quantification of information for dark versus, say, well-studied mode of action drug targets, and ask the question, if I were not to rely on biomedical literature, are there any information channels that could provide me enough information to prioritize them for biomedical
Well, yeah,
research?
that's so interesting, Tudor. So, 20,000 proteins or so, 600 of them are commonly used, and underneath that, there is 500 that could provide fairly high utility. And so if I understand this correctly, Tudor, are you saying the biopharmaceutical industry is really focused on the 600?
Jill, that's a good question. I wouldn't say that. To some extent, of course, it is safe to say that a lot of the pharmaceutical companies follow what's known in the industry as the Me Too model. And the Me Too model is essentially as soon as somebody comes up with a drug on the market, then other companies will rush with similar products to compete with the previously approved drug. And that per se is not a bad thing. If you think that until recently the best-selling drug in the world was Lipitor, Lipitor was the 7th drug of its kind to come to the market.
Yeah. Yeah.
So it takes a lot of tinkering. It takes a lot of tinkering, a lot of improvement to create what would be considered the best-in-class drug. So to that extent, it is difficult for the pharmaceutical companies to come up with new drug targets, and that's really the difficulty here. It's out of 600 proteins, the reason we only seen an increase of about 10%. So in 2013, when I did first this analysis, we had about 600 proteins. Now we're about 660, including drugs that were approved in 2019. And what that means is we had roughly a 10% increase in the number of drug targets over 8 years. To my mind, that's a rather conservative approach because it's so difficult to get regulatory approval and put new drugs on the market.
Right. So obviously these companies have sort of a risk management process, and the economics could be quite different when you go for first-in-class type compounds and targets. So, is your attempt here is to make that 6,000, 6,500 or so what you call the dark genome proteins, but they bring them to the fore, so to speak, by combining data from various avenues?
Yes, correct. So my hope is to draw attention to the untapped potential of the understudied proteins and sort of bypass the process by which a protein is studied, say 10, 15, 20 years before somebody considers it a valid target for drug discovery. And in that sense, I look at data that comes from genome-wide association studies. So GWAS is a channel of data. Another channel is the International Mouse Phenotype Consortium. And then a third channel that we're looking at is a combination of all these information channels with the prioritization scheme that I mentioned, which divides them into the 4 categories.
Yeah, so this database that you mentioned, Tudor, Pharos, is that what it's called?
Correct.
So how does that data look like? How is it organized? Just very, very at the very highest level.
So Pharos is the platform where the data is accessible, and we've actually just published a paper that I also sent you a link about, which is called TCRD and Pharos 2021, Mining the Human Proteome for Disease Biology. So with your permission, I'm going to switch in this context to that paper. So essentially, that database, TCRD, catalogs information from about 75 different individual sources, and it looks at information from a number of sources or channels, for example, related to protein expression data. So we have essentially major categories of data, for example, expression data. Another type of category is what we call protein-protein interaction data. And the newest addition to that in 2020 was to include virus interaction proteins. So there is a atlas that was developed at Columbia University, which is called pHipster. The first author of that paper was Gorka Lasso, but it was work done in the lab of Barry Honig. And essentially, pHipster looks at the interaction between viruses and human proteins, or viral proteins and human proteins. And in the context of the current pandemic, we thought it relevant to add that information channel.
Okay. So it's a large amount of very interesting data. So are you pre-processing that data to some level, applying some AI techniques for on?
We certainly pre-process the information. We try to normalize it, and then we try to store it in what is commonly called machine learning ready format. And so we try to standardize it, observing certain ontologies. So we look at all this small molecule data, protein data, disease information, genomic, phenotypic, and ontologies. But I'd say the only part where we currently use some artificial intelligence is when we do text mining.
Yeah.
Because text mining, and that's work developed in the lab of Lars Jensen at the Novo Nordisk Foundation Center for Protein Research in Denmark. And so Lars uses some machine learning tools not only to process PubMed abstracts, which is a lot of text mining technologies do that, but he also processes the full text publications available in PubMed Central. And he uses BioBERT, which is a two-way language, bidirectional language encoder. And the other thing that he's doing, he's using what's called named entity recognition. So if you have an entity called a disease and another entity called a protein or a gene, then the question is, can you establish associations between proteins/genes and diseases, or phenotypes, as it were? And that's where we use some sort of artificial intelligence precursor or machine learning, I would say.
Are the users typically from an academic institution or pharmaceuticals and biotechnology companies are using it as well?
We offer the database for full download. So because of, I would say, confidentiality issues, what most likely is happening with respect to biotech and pharma industry is that they download the complete dump of the database. And there is also a Dockerized version of Pharos. So they essentially can install behind their own firewall a version of Pharos complete with everything that we have. So Pharos, the front end of the system, uses GraphQL and quite modern query systems to enable the users to rapidly access information. As I mentioned, 75 different sources of information that come together into a single web page.
Yeah.
And it's using a dynamic query system. So, for example, if you have a protein that's very poorly studied, it will not have widgets or empty spaces where whatever data is missing, it would just say, I don't know, drugs, and there will be no drug available. So it will simply not show you that tab which has the drug part. It will only show that for proteins where drugs are available.
Right, right. Yeah, I mean, one issue with that, though, is that since there is no sort of two-way communication, if the industry, if the companies are just downloading the data and then use it privately, they're not really sharing any information back, right? And so this has always been an issue. And so, is there any attempt to try to make this more generally available from a two-way communication perspective? Obviously, there are IP issues and privacy issues, but do you think NIH or other agencies might do that?
Jill, I absolutely like your idea of the two-way communication in this particular aspect. Of course, intellectual property becomes an issue, so it's really a question of do companies, are they in the position where they'd be willing to share? And obviously, the most clear way to communicate that type of information is to place it in a publication where we just download the supplementary material. If they were to, say, be interested in contacting us directly, we have that information available. So in the FAQ section of Pharos, we pretty much say, here are ways for you to contact us. And if somehow somebody says, we have this really large set of data we would like to contribute to Pharos, we would definitely consider including that information.
Okay. I want to touch on one other, this is a different thing, right? So Drug Central 2021 supports drug discovery and repositioning. And that's another resource, drugcentral.org.
Correct. So part of the reason we catalog proteins across into 4 different bins is that the most studied ones that one would consider pretty much the textbooks of biochemistry and pharmacology are based on are essentially the drug targets.
Yeah.
But the only way to understand drug targets is to map drugs onto their mode of action targets. So we started that process about 7-10 years ago, and we accumulated this database that we currently call Drug Central. And essentially, Drug Central, which is also available just like Pharos for a complete database dump, and there are ways to get a Dockerized version. So if anyone is not comfortable using the system as is, they can install their own version in-house.
Yeah.
Essentially, what we do is we catalog information about their chemical structures and makeup, so pretty much what's in the pill, and with information about the dosage. And this is what's called active pharmaceutical ingredients. Sadly, we do not catalog the inactive pharmaceutical ingredients. That would be a subject of perhaps future work. But then we look at all these drugs, and then we map them to drug targets. We also map them to diseases. And when I say diseases, I'm talking about drug indications. contraindications, and off-label medical uses. More importantly, and I think we are the first to offer this in the public domain, we catalog side effects by sex. So we separately index information for men and women with respect to the drugs. We take that data from an FDA system called Federal Adverse Events Reporting System, also known as FIRES. So we process that information. And of course, we have additional data available about drugs.
Okay. Tudor, we'll take a quick break. When we come back, we'll talk about drug repurposing, some AI techniques, especially in the context of what we're dealing with today, the COVID-19 problem.
Okay.
Thank you. This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com. So we're back. Tudor, we were talking about data coming from various avenues that could put a better focus on the large number of proteins in the human system that could be utilized from a drug targeting perspective. And you've been collecting and organizing this data. There is one from NIH called PHAROS that is publicly available. There is also drugcentral.org that has been organizing existing drugs. The properties and the effects, again, for use in perhaps in machine learning and other methodologies. And more recently, you were looking at some of this from a COVID-19 perspective. So you have a paper that came out. It's entitled Virtual and In Vitro Antiviral Screening Antiviral Screening Revive Therapeutic Drugs for COVID-19. And you are looking at existing drugs, right? Thinking about repurposing those drugs like hydroxychloroquine.
That's right, Jill. So thank you for having me back. So in addition to these NIH-funded projects through the Illuminating the Druggable Genome, which include PHAROS and Drug Central, we were considering how to best put to use these technologies in the context of the COVID-19 pandemic. And in that sense, we started with hydroxychloroquine, and then we asked the question, if we were to start with hydroxychloroquine as a potential antiviral, can we find an antiviral that is better than hydroxychloroquine and potentially has fewer side effects? So that was the subject of this paper that you mentioned. I should perhaps introduce the listeners to the concept of virtual screening first.
Yeah.
So the idea behind virtual screening is that you do a in silico study. So this is purely computational. And there are two ways to do this, called ligand-based virtual screen, and the other one is structure-based virtual screen. And in the first case, the ligand-based, let's say what you're looking for is a car. And you pretty much have a model of a car, say a Volvo SUV, the XC90. And then you are asking the question, there are all these thousands of other cars out there. Some of them will, of course, be coupes. Some of them will be pickup trucks, etc. But the question will be, if I have this template, which is the XC90, which other car looks most likely to be similar to the XC90? And of course, there are criteria that we defined, like it has to have four wheels, it has to have an engine, a windshield, all these other things that come as intrinsic properties. These are properties that we can compute for drugs. They relate to the shape of the drug, the electrostatics, the charges, all sorts of properties that we can calculate. So that is what we call ligand-based virtual screening. The other one is structure-based virtual screen. So the drugs, the way they work inside the body is that they interact with our proteins, or what other people would call receptors. And for the drug to bind to a receptor, it basically has to fit inside a binding site or a cavity. So think of it as a car going into the garage. So what you have to find is an appropriate garage for your car. If the garage is too large, like a hangar for the, Hercules C-130, then obviously that's not a very good fit because your Volvo can fit anywhere. If it's a very tiny garage, like a toy house, obviously it's not going to fit either. So you have to have the right garage and the right car, right? So that's what structure-based virtual screening does.
Okay.
In this case, and in this particular paper, we did what's called ligand-based virtual screen. We started with chloroquine because at the time we considered chloroquine to be a good starting point because it had activity in vitro. So there is a particular assay called the cytopathic effect. This assay, what it usually does, there is a particular cell line called Viro E6, and you infect the Viro E6 with a live virus, in this case the SARS-CoV-2, which causes COVID-19. And then you see whether this drug can protect the cells from dying because they are killed by the virus. And so hydroxychloroquine does have that effect that against Viro E6, it protects them from this cytopathic effect at a pretty good bioactivity. It has a pretty good bioactivity. What we discussed in the paper, after multiple iterations, and this was key in our publication, we did not rush to publish. This was not a preprint. What we did was we found some observations and experimentally we identified some drugs, which include the antimalarial amodiaquine, a anti-blood, how should I say, a drug that protects against high blood pressure. This one is called nebivolol. And last but not least, an antipsychotic called zuclopentisol. And these three drugs, we found them at the University of New Mexico in the lab of Stephen Bradfute. They were found to be active against the virus in this cytopathic effect assay. So what we then did, we purchased these drugs from another vendor, so a chemical vendor. You know, you can buy these chemicals just as you were be able to buy a car, just from different car dealers. So we bought from a different chemical manufacturer, and we sent them to the University of Tennessee in the lab of Colleen Johnson, where she did different experiments. But they all came to the same conclusion. These three drugs are active. But what's equally important, at the University of Tennessee Health Sciences Center, Colleen also performed a different experiment, which is called a viral titration assay. In the viral titration assay, what you do is you progressively expose the cells to a different load of virus. And so you pretty much try to ask the question, how well does the drug protect against the virus? And it turns out that this drug called remdesivir, which everybody uses, and it's the only FDA-approved drug specifically for SARS-CoV-2 infection, is very active, but equally active is this amodiaquine, which is an antimalarial drug. However, hydroxychloroquine has no activity.
So the idea here is to look at existing drugs and existing drugs' activities, perhaps their mechanism of action, and come up with a combination of them. How exactly would you ultimately create something that might be useful for COVID-19?
That's right. The thought is first to identify drugs that have some activity, in this case, nebivolol or amodiaquine. And then the question is, can we combine them And we haven't yet finalized that work. And part of the reason we haven't completed that, we sort of expected that by this time, like mid-December, we should be had completed that work. Turns out that 5 of the lab members from Colin Johnson's lab had tested positive for COVID-19 due to, this is not infection at work, this is literally society type interactions. So parents or contacts in their immediate contact network had tested positive. So we pretty much had to slow down the work. They are now back in the lab. They hope to restart the experiments soon. So I really wish them good health, and I hope they can get on with work. It is very important what they do, of course.
Yeah, so you are actually using some of the data that we talked about before, both the Faro type data, but more importantly, perhaps the drug central type.
Correct.
And so you have another paper. It's entitled "Artificial Intelligence, Drug Repurposing, and Peer Review," and they're kind of interesting things. So you could apply machine learning techniques to sort of custom design something because you could predict potentially because you have some understanding of the toxicology, toxic effects of existing drugs and their structure, and perhaps that allows you to assign some probability what a new combination, what type of toxic effects a new combination might have. Is that the idea?
So the paper that you particularly mentioned, it's a commentary in Nature Biotechnology, and this was spearheaded by Jeremy Levin, who is head of Ovid Therapeutics, which is a New York-based company, as well as Alex Javronkov, who heads a company called In Silico Medicine, which is an artificial intelligence based company. And essentially what we tried to state in this commentary, and we also recruited assistance and insight from additional folks, including AI-based companies in Israel and in France, particularly Spark Beyond and Aukin. So Sagi Davidovich and Thomas Klozel are the people I'm having in mind. And what we try to address in this paper is the question, can we deal with the deluge of information that comes to us via preprints and peer-reviewed publications and TV channels and Twitter, which all claim to have solved the problem of COVID-19 and they have found a cure. And alas, we know that that's not the case. Just because somebody publishes on Twitter doesn't constitute scientific results. So what we try to do is point out that we really need to be more aggressive in trying to call out fake science for what it is. So the paper starts with the sad situation caused by Andrew Wakefield and colleagues who published in 1998 a paper that linked vaccines to autism. This unfortunately has started a movement called the anti-vaxxers. Right. That paper has been retracted, but it took the Lancet 12 years to retract that paper. So at the time, we were literally concerned that, for example, with hydroxychloroquine and chloroquine, we saw a very aggressive behavior from the White House and others in France, for example. They were stating that chloroquine and hydroxychloroquine can block the infection. That is not the case. I think everyone can relax on that. Scientific evidence points to the contrary. But at the time, we were concerned that if this does continue unchecked, it ends up in people taking the wrong medication for the wrong disease. Because to be clear, hydroxychloroquine does have its role in autoimmune diseases and in malaria. It's just it shouldn't be used in COVID-19.
Yeah, so this is an important point. So the pandemic actually taught us that if you are not prepared for it, it's going to take some time before medications or vaccines going to be developed. And if it's a worldwide pandemic, there is going to be a lot of misinformation. You mentioned here, according to a recent analysis, an average of 367 COVID-19 papers are being published every week, with a median time for submission to acceptance of just 6 days. And so that is potentially a prescription for disaster. And so do you foresee some sort of an AI-driven system in place to either complement or potentially change the peer review process?
Yes, that's pretty much what we're hoping, is that given appropriate funding, such a system can be developed. And of course, it would be impossible for an AI-based system to read 300 papers and decide which paper is right and which paper is wrong. What this artificial intelligence system could do is identify conflicting information that would be automatically flagged for further human intervention, and we would also identify things that by imputation, by inference, would be in line with previous observations based on the existing corpus of biomedical literature. So an AI-based system would have the ability to not only distill all of PubMed, which is 30 million publications at the abstract level, as well as, of course, the 4 or 5 million that have full text, but could also look at the patent corpus or the clinicaltrials.gov data, and then ask the question, what people claim in this new publication, could that publication be in line or against the current consensus based on these text corpora? And of course, if there is a consensus and this paper is in agreement, it just extrapolates by a little bit, then it's one way to flag for peer review. If it would say this totally contradicts what we currently know, then it would indicate that either there has to be very solid evidence and multiple confirmations that this is the case, or perhaps this paper is just plain wrong.
Yeah, I mean, it's a bit analogous to fact-checking the politicians, Tudor. So what you're, if I understand this correctly, you have a compendium of historical information, drugcenter.org, clinicaltrials.gov, and any new paper could be analyzed in the context of that compendium of information. And if there is some sort of an AI process running, it could at least assign a probability that the conclusions of that paper are likely to be correct. And at the very least, it will classify the output into buckets. And so, especially in a situation that you need fast information to solve something that is really accelerating, it would at least focus both the scientists and the policymakers into a subset that is more likely.
That's correct. So what we're hoping to accomplish in the next 2 to 5 years is to develop a system that would pretty much assist the peer review process with a set of predefined rules, which would then help triage the papers into likely to be true or unlikely to be true, or somehow measure the element of surprise that is brought by the paper. And what I mean by surprise is, of course, not in line with what was previously known. Again, I will emphasize that mathematics, unfortunately, does not help detect truth from falsehood. That's just currently not possible as far as I know. But when it comes to biomedical literature, at least we can reasonably state this is in line or this is not in line with what was previously thought to be true.
Yeah, I mean, obviously, the issue with biomedical research is that there's a lot of uncertainty involved, and so it is not really a deterministic, sort of mathematical process. On the other hand, it also creates problems for humans, right? So, one could argue peer review could go into confirmation and confirmation biases. And so humans are not unbiased entities either. And so
That's correct. We all know of cases where groundbreaking work that eventually got the Nobel Prize was rejected for years because it didn't fit with existing dogma. At the same time, we should also be aware of retraction watch, where people simply fake the information that gets into the papers. And then the other one that I don't even know where to categorize are the paper mills, which is a new phenomenon where papers that appear to be true are pretty much invented out of thin air. And they all fit a certain pattern. Hopefully, you should invite someone to talk about it to your audience. But such phenomena exist, and what we're hoping is that over time we will accumulate enough evidence to detect them. Unfortunately, as you identify the elements that detect them, the people who create these fake papers also get smarter and will find ways to beat the system. So I'm sure this will be a cat and mouse game for a while.
Yeah, that's a good point, Tudor. There are technologies that are coming or already available, GPT-2, GPT-3, and these technologies are very highly sophisticated. So given raw materials, perhaps existing papers, the technology could write a scientific paper that could be read, to be very good, actually. So to find fault with it requires a lot of effort. And so...
It certainly does. There's a lot of detective work involved.
Yeah. So this is probably what you mean by paper mills. And that problem is going to only increase now and only get more sophisticated. And so the question remains, like you say, cat and mouse game. Both the volume and the sophistication of information is going to increase, and to parse what might be useful and what might be just either useless or just a framing. I think it's a major problem. And I don't know who would take the responsibility for this, Tudor. If you think about it from a policy perspective, is it social media? Is it organizations like NIH? Who would be ultimately responsible for implementing something like that?
So to some extent, of course, NIH has become well aware of this. If I remember correctly, Francis Collins addresses in one of his commentaries the issue of reproducibility, which is pretty much the crux of the issue here. And then there are the so-called data forensics detectives who examine publications and try to come up with the possibility that somebody has pretty much taken information and faked it, like images. Yeah, they process images with Photoshop, something like that.
Right.
So the name that comes to mind is Elizabeth Bick. And she does a lot of work in scientific integrity. And speaking of scientific integrity, a shout out to Anita Bandrovsky and her work, the so-called size score. So there is a company that initially this work was funded by NIH, NIMH. They funded this work. So if you go to sizescore.com, you will find something called the Rigor and Transparency Index, which is an effort to try to evaluate transparency and rigorosity of publications. So I think over time, if SciScore really takes off, we will be able to evaluate the degree of confidence in publications over time.
Yeah, so Tudor, in conclusion, if you look forward 5 years, where do you think we will be? There's a lot of data being generated. You and others are trying to aggregate and analyze, pre-process, stage that information. AI techniques are improving. Where do you think we will be 5 years from now in the general area of drug discovery and repositioning?
In the drug repositioning component, I think in silico technologies that are really rapidly developing, I think it's sort of I don't like to use cliches, but it's almost a no-brainer given the pandemic that we really need to invest a lot in smart automated technologies, perhaps robotics-based science to be able to conduct experiments without people being present. I think that's really up and coming. So there will be robotic labs that will carry out, say, antiviral research while the people are working from home because of lockdown orders. So I think that's entirely within the possibilities of what's coming in the next 5 to 10 years. On the drug repurposing side, the more we understand the way drugs work, the more likely it is that we'll be able to identify novel clinical uses for existing drugs. And that's really the crux of drug repurposing. And when it comes to artificial intelligence assisting scientists, I also fear that we humans will simply not be able to read the millions of publications. And I'm not just thinking about the publications related to COVID, but say you're a neuroscientist interested in Alzheimer's disease. I don't think there's anyone out there currently being capable to read and comprehend all the publications that are coming. And we're not talking about the papers that are fake. We're talking about legitimate papers. The number of papers that are published in each area is dauntingly increasing. So what I expect to happen is that there will be either phone-based apps or some sort of system that's rooted in something similar to what Google is developing with Google Patents and Google Scholar, which will then be able to take a subset of these publications and distill them into a resume or a summary of those publications and feed them to you as a scientist. So you will pretty much have what currently in science is known as reviews, but generated by a machine. So I also think that's up and coming.
Yeah, it's exciting times. It's almost like it's a necessary condition for us to internalize information that is exponentially increasing. So
yeah, we need to get smarter in getting smart. That's what I would think. Yeah.
Yeah, excellent. Yeah, this has been great, Tudor. Thanks so much for spending time with me.
Thank you for inviting me, Jill.
Thank you. Bye.
Okay.
This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com.